Well, thank you so much. Good evening, everybody. It's really a huge pleasure to be able to speak today. Thanks to Annette and Charlene for organizing this lovely event. Last year was my first attendance at a patient information session, and it was a great time. Sorry? Oh, you can't hear me. Sorry. Okay, there we go. So anyway, it's a pleasure to be here tonight to talk to you about some of the research that's going on in Montreal. To try and tackle some of the problems and challenges in this disease. So I'll give you a quick overview on general information about melanoma, and then I'll dive right into some of the research efforts that have really led to the development of novel therapies in treating metastatic melanoma. And then I'll tell you a little bit about some of the challenges, clinical challenges that exist still in this disease, and how the future really is bright for overcoming these challenges by doing medical research. So what is melanoma? Melanoma is a cancer that, is there a pointer in it?
I'm not sure that it works, Sonia. So.
It's fine. So melanoma is a type of skin cancer that has its origins in melanocytes. These are the melanocytes here. Whoops. There. Sorry. And so melanoma is considered the deadliest form of skin cancer, and this is because if it's left undiagnosed or untreated, then it has a tendency to spread very quickly throughout the body, and this is referred to as metastasis or metastatic disease. And so having said this, that melanoma can be extremely deadly, it's also highly preventative. So there's a high survival rate for patients diagnosed with melanoma if the disease is caught early, as Ned alluded to in her introduction. And so I always include, when I give a scientific lecture on melanoma, the ABCDEs of recognizing melanoma. And so if you have a family member or yourself or a friend, it's always nice to have a look and see if you can spot the melanoma by if there's a mole that looks asymmetrical, if the border is uneven, the color is patchy, if the diameter is larger than that on the head of the eraser on a pencil, or if any of these characteristics are changing or evolving, then for sure you should encourage yourself or your loved ones to go and seek a dermatologist for a diagnosis. And so melanoma can come in various flavors. You have cutaneous melanoma, which is the most common type of melanoma, often presenting on the torso in males. But melanoma can also occur on other sites of our body, in the eye, uveal melanoma, on the palms of the hands or soles of the feet, and this is referred to as acral melanoma. Acral melanomas are not commonly present in Caucasian populations, more so in Asian populations and people with darker skin. And melanoma can also occur on mucous membranes, such as the oral cavity, and that's called mucosal melanoma. So these researchers have really had a big influence in telling us a little bit about the underlying genetic causes for these different types of melanoma. In the case, for example, of cutaneous melanoma, we really have a very good understanding of how harmful UV light can cause changes, permanent damage actually, to the DNA of skin cells. And so what happens is the UV rays will hit the DNA at specific sequences, and these specific sequences are called genes. These genes then acquire a mutation, which is the permanent damage in the DNA. Usually the gene is encoded by a series of letters. I have an example there, ATGCAC, and oftentimes what happens is there's just one letter change, and so this then causes a faulty gene or a mutated gene. And so the consequence of this can be quite severe. So if you have a skin cell that's acquired a gene mutation, this will encode for a faulty protein, so a protein that no longer has its normal function in the skin cell. And so a protein is something that does the job in a cell. So it responds to signals, and it tells a cell when to start growing, when to stop growing. And so whenever I talk about mutations, I try to use the analogy of writing a children's story. So for example, if I wrote a story about a child kicking a ball and I had a typo in the A, then the child would kick the ball. This would have very serious consequences for the remainder of my story. So if we apply this analogy to the most common mutation in cutaneous melanoma, which is the BRAF V600E mutation, what happens is you have a change from in the gene from GTG to GAG. So perhaps some of you have been diagnosed with melanoma or one of your loved ones, and you've been told that your melanoma is BRAF positive. This is usually how it's diagnosed. You have your genes sequenced, and you find out, the doctor tells you that you have BRAF positive melanoma. And so what does this mean for a normal skin cell? Basically, in a normal skin cell, this BRAF protein is essential for regulating normal growth. When you have this V600E mutation, the cell no longer cares about external signals that are coming into it. BRAF just tells this cell to grow out of control, and you get uncontrolled growth, and this causes melanoma or skin cancer. So why do you as patients, or me as a scientist, care about the mutational status of your melanoma? This is in large part because by understanding the function of this mutated BRAF protein, scientists have actually been able to develop new therapies for melanoma, more personalized therapies for melanoma. And so this then brings us to talk about targeted therapies. And I'm not going to go into depth on this slide because Wilson, being the clinician in the room, is going to talk at length about the benefits and challenges of these therapies. But all this to say that research has really driven the development of these molecules or these drugs that can inhibit the action of this faulty BRAF protein. And a second type of targeted therapy, which are called immunotherapies, are basically using the patient's own immune system to survey, recognize tumor cells, and kill those tumor cells. And those are referred to as immunotherapies, and there have been a lot of success stories with immunotherapy. Stan in the room will also be talking to us later tonight about his experience with those drugs. And so although there has been a lot of success stories with both types of targeted therapies, clinical challenges still remain. So sometimes with the BRAF inhibitors, you have, they just stop working, and so the disease comes back, your melanoma comes back, and this is what's referred to as therapeutic resistance. In the case of the immunotherapies, although yes, there have been very impressive results in some patients with long-lasting remissions, the fact is really only 20 to 30% of patients will respond to these immunotherapies. And so clearly these are challenges, clinical challenges that need to be overcome, and it highlights the need for more research. And so just last month, the melanoma research community received an early Christmas gift in major funding for melanoma research in Montreal. Basically, we were able to set up, and Wilson and I are part of this initiative, we were able to set up a Montreal Cancer Consortium, and the focus of this project is really bringing together 7 of the top universities and hospitals across the island of Montreal to band together, so scientists and clinicians all banding together to try and tackle some of these big clinical challenges in melanoma. And so the overarching goal of that project is really to improve treatments for patients with melanoma, and with the forward vision of perhaps applying some of what we find in melanoma to other disease types. So the focus again is on targeted therapies, and the targeted therapies that I just mentioned. some of the questions that the project has been funded for is to, for example, can we develop new therapeutic combinations to overcome targeted therapy resistance? And this is a project that's close to my heart. MINC1-2, we discovered these are two proteins. MINC1 and MINC2 are two proteins that we discovered are a novel therapeutic target in melanoma, particularly acral melanoma. We published that study last year. And for acral melanoma, there really aren't very many good therapies out there. And so what we're trying to do in the lab is to try and make MINC1-2 inhibitors work better. And this is the project of Sethan Prabhu, who's a PhD student at the back of the room. So he's been tasked with trying to figure out what drug combinations would increase the effectiveness of MINC1-2 inhibitors. We've been working with pharmaceutical companies to develop better inhibitors, and we're very humbled to have received funding from the Melanoma Network of Canada to support some of Seth's beautiful work. So again, we thank Annette for putting forward our application and for providing funding for this study. And so one of the other big questions that we're trying to ask via the Montreal Cancer Consortium is, can we predict which patients will respond to immunotherapy? So I told you already that really only 20 to 30% of patients will respond. And one of the challenges is, which patients will respond just to single agent immunotherapy versus combination agent immunotherapy. So there's different flavors of immunotherapies. And now there are a lot of clinical trials going on, which Wilson will tell you about, Dr. Miller will tell you about, where you're combining different types of immunotherapies that have different impacts on immune cells. And so although, again, these immunotherapies have been a splash all over the news with patients, their disease just melting away and being in remission, sometimes with the combination immunotherapies, you have some very toxic side effects that can happen. So the cost of your melanoma going away maybe comes at a price of having some very serious, at times, immune adverse related events. And so there too, researchers are doing their best to try and overcome that clinical challenge. And this is a study, the Cancer Immunotherapies and Immune-Related Adverse Events Study, big long title, for really just trying to predict which patients will be at highest risk to develop these toxic side effects from the immunotherapies. And if we know that they're going to be high-risk patients to, have problems with their lungs, their liver, colon, then we won't put those patients on these combination immunotherapies. And so this is a large study, again, that we are trying to get funded for. Wilson Miller and his team in the Clinical Research Unit are heading this study along with Dr. Marie Hudson, who is an expert in understanding and treating patients with autoimmune diseases. And again, this is a multi-hospital study really aimed at improving cancer treatment for patients diagnosed with melanoma. So I hope with all of this, you guys walk away with knowing that there are people out there in hospitals and research centers that are really trying to make a difference at improving care for yourselves or your loved ones that have been diagnosed with this disease. And so I'll just plug again, Annette already introduced the Strides for Melanoma walk that happens on our beautiful Montreal in September. We do have a team that will be walking and running. Our team is called the Skincredibles, and street cred to Seth for coming up with the name. So for anybody that would like to join our team or consider a donation to the team, you can find our team on the Strides for Melanoma Network website. And so with this, I'll close my acknowledgements. All the members of the Miller del Rincon lab, without them, really, they put in so many hours in the lab at trying to, do the research and discover something new. I mean, it's a lot of struggles sometimes in the lab, but really when you discover something novel, it's just so amazing, and to have an impact on patients' lives, even the pinnacle of doing basic research. And then we have collaborators across the island of Montreal, the Goodman Cancer Center, Hôpital Maisonneuve-Rosemont, Centre de Recherche du CHUM, McGill University Health Center, and then the Melanoma Network of Canada, of course. And a shout out to François Lagacé for helping put out the promotional materials for this patient information session. Thank you very much for your attention.